Okay, so we're going to introduce Chapter 13 now. To remind you, we're not going to get through all of Chapter 13. We'll get through quite a bit. But the cardiovascular system is going to span the third and the fourth unit. Maybe we can't get it all today. It's complex. And so what we're going to do today is Objective 1. Look at the components of the cardiovascular system and give a brief description of the function of each. Keeping on the brief part, we'll expand that when we get into Unit 4. We're going to look at the major structures of the heart, which if you've been in lab already, you've got to see some. And then we'll review the flow of blood through the heart like we studied last week in lab. What will be new to us and aligning with our lab this week is the trace of action potentials actually through the heart, how they physically go through the heart. If we have time, we'll talk about the cardiac cycle, tie that into the electrocardiogram, also tying into the lab this week, and then kind of mush everything together to make it hopefully make sense. We'll also review the intrinsic versus the extrinsic control of the heart and go into better detail how our autonomic nervous system can directly influence these things called afterload, preload, and contractility. So we'll get into more detail about how the nervous system affects our heart. So the first thing is we want to just look at the cardiovascular system. There are three primary components which comprise this system. And the most famous would be the heart. You're also familiar with your blood vessels and blood. These three structures and systems, I guess tissues really, will make up the entire cardiovascular system. The heart, we're going to introduce obviously here in Chapter 13 and pick it up in Unit 4. Chapter 14, we'll talk about our blood vessels. And then Chapter 15, we'll talk about blood. So we'll span the 2 units. What do you think the function of the heart is? To pump blood. It serves as a pump. It pumps and distributes blood. Let's acknowledge as well about the heart that because it pumps, it sets a blood pressure gradient. It establishes, maintains a blood pressure gradient. We've used that word all semester, gradient. Blood pressure gradient must be then, it's a blood pressure gradient.
Like the pressure?
Yeah, you just pick it apart. What's a gradient? Higher in one and lower in another. Very good. So gradient means more to less, or whatever, there's a difference. So a blood pressure gradient must mean that
there's more blood outside the heart.
There's more pressure.
let me restate it this way. The heart establishes a blood pressure gradient. So it tells us
Pressure is more in one place than less.
pressure is more in one place than it is in another. Because we want to do what with the blood?
Get it all around the body.
We want to pump it, we want to move it. Which way in respect to the gradient? Down the gradient. From a higher pressure to a lower pressure. We want to send the blood out, and we want it to come right back, don't we? So we can do it again. Yes?
Are we saying that gradient and potential are synonymous?
No.
No. Because one has to do with electricity or current.
I would not say they're synonymous. You could use the word gradient to describe a potential, but I would not use the word potential to describe. You could work it that way, but I don't want you to confuse action potential with gradient. So the heart establishes a blood pressure gradient, right, by pumping. We'll look at the pressure. We'll actually look at real values when we get into the circulatory, into the blood vessels, and then review them in the respiratory system as well. We'll look at that pressure gradient. Another important feature of our circulatory system is the presence of our blood vessels. What is their job?
Take the blood to the
Transport the blood. Transport. It's actual physical passageway, if you will, for our blood. So it takes takes blood to its destination and back from its source.
Are they kind of like live pipes?
Sure. They're kind of like pipes, but yeah, they're not dead. They're alive. They're cells, tissues, organs.
They don't do any things.
They do all of that you're just doing.
Well, I know they can expand, but they don't have to have a motion.
Our blood vessels, some of them do have muscles in them, which can contract. And so that we will study in chapter 14 in the next unit how that, if a blood vessel contracts or not, changes our blood pressure significantly. Okay? And so yes, it certainly can. So our blood vessels are there to distribute our blood from a locale to another. There's going to be different types of vessels. They're going to be built differently. They're going to function differently. So we'll clarify that after the test. And then we'll talk very briefly about maybe the most important, I don't know, type of hierarchy, but a very important component of our circulatory system is actually the blood. And so we'll talk about the components. It's not just red blood cells. We now know there's water in there, and there's hormones in there, and neurotransmitters and nutrients, okay, and immune cells and proteins, and keep going, right? So we'll talk a little bit about the blood. These are our three primary components of the cardiovascular system. Are there any questions before we look at the heart in more detail? If we were to try to anatomically find the heart, it's in what you would call the thoracic cavity, behind the sternum, and it does tilt a little bit to the left. So when you go do the Pledge of Allegiance at the baseball games this summer, you're putting your hand over the left side of your chest because you're covering your heart, right? So it's kind of where the heart is. It sits above the diaphragm, which is above the abdominal cavity. Okay, so it's just right there in the middle of the body.
They should have smacked Daddy in the middle, huh?
Goes to the left a little bit.
That's why the left lung is shaking when you see it.
Yeah, the left lung will be built differently because of the heart projecting two lobes. That's correct. We'll notice when we get into the respiratory system that the left and the right lungs are built differently. One is accommodating, the left lung is accommodating that heart. All right, so it's there. Of course, we're going to go way more detailed than that, aren't we? Good old physiology. So if we look at the anatomy of the heart, let's review. How many chambers does our heart have? 4. We have a 4-chambered heart. The upper chambers are called atria. The bottom chambers are called ventricles. Very good. All right. So I'll just kind of walk through. I have labeled here the right side. Now remember, you're looking at a patient. It'd be the patient's right side versus the left side. How do I know how it's cut? There's a couple of clues to look at.
The left ventricle is a lot thicker than the right one.
The left ventricle is thicker than the right because it does pump to the systemic circuit. Very good. We have some color hints here as well. The right atria receives blood from which circuit? Systemic circuit, so the blood oxygen level is a little bit low. Those are usually identified in the blue vessels. Superior and inferior vena cava. What else? If you're really getting anatomy, you could look at.
It has something that the left ventricle doesn't.
The aorta, the right ventricle, the papillae muscles versus. You'll hit that in anatomy. The big things that I like to focus on is the thickness of the left versus the right. And then yes, if you do kind of scrutinize, you can see that this vessel's pushing blood out to the aorta, which has a really characteristic shape. So if this is connected to the aorta, this must be the left ventricle. That leaves us 50, right? Okay. And above that would be the right atrium. So the right atria sits above the right ventricle. And then over there on the other side, we have the left atrium and the left ventricle. In between, let me see what else I've labeled. The blood vessel that drains blood to the right atrium is called the vena cava. And some of you guys are aware, you've been in anatomy, you have one that comes from the top and one comes from the bottom, so one would be superior and the other would be inferior vena cava. We also identified the aorta, which is the large artery that takes blood out to our systemic circuit. Additionally, there are unique valves in the heart. There are valves between the atria and the ventricles. Hence their name, the atrioventricular duet. You could look at the tricuspid and you could look at mitral. I always get their names backwards. I just AV valve, semilunar valve. I stick with those because I can handle that. So help me out. Tricuspid's over here. Is that the AV? It's got the three attachments of, you call it the mitral?
Yes.
Bicuspid. The aortic valve, what do you call it?
Aortic valve.
I'm on it.
There's a
And then the
I forget what the
pulmonary
word
valve.
is, but it's like TPMA. Tricuspid, pulmonary, mitral, aortic.
I will never forget now. Thank you. I gotta stop blushing now. I'm now gonna call the tricuspid valve because I've got to pull it. Yeah. It works. Even I can remember it. Before a moment ago, I used to call this just the AV valves, atrioventricular valves, because of their location, the atria and the ventricles. The first one does have three, right, connections. And if you're in the lab already, you got to look at the actual heartstrings. If you're in lab today, we may not have time to do the dissection. We'll see. We are going to have that guest speaker. So anyway, so we have the AV valves between the atria and the ventricles, and the semilunar valves between the ventricles and the respective circuit. All right. And then, yes, they would have their appropriate name to which circuit they're going to. The pulmonary valve would go to the pulmonary circuit, and the aortic valve would go to the aorta. What do our valves do? What is their job?
One-way blood flow.
Prevent blood from going backwards. Yeah, very good. What I want to check your terminology, because some people will just inadvertently just say the valves contract. And it's like, oh, the valves will contract, and I'm going to say, and why would I do that?
Because they contract for a reason.
They don't contract.
I mean, they
They
close.
open and close.
Open and
muscles contract, which opens and closes the valves.
Oh, muscles open. Muscles will pull or push on them, but the valves themselves aren't muscle. So it's not muscle. It can't contract. So it's just like a door. Right? If I were to get technical, if this were my valve, right, the door itself doesn't open, right? I would be the muscle. I would be opening the door. I would be shutting the door. Does that make sense? The valve itself is just kind of responding, doing what it's told, if you will.
Well, I was
That's
just,
what all the.
I don't know if this is right, but when I thought about it, I was like, when the atrium gets full, the pressure.
Yeah, and they are driven by pressure. Okay, and right. So pressure predominantly is what opens the semilunar valves, but pressure and muscle activity opens up the AV valves.
Okay.
But certainly, remember, our heart establishes a pressure gradient that also pushes our blood from one locale to another, allowing those valves to open or close. So when the pressure gradient reverses, let's say I'm in my right ventricle, the AV valve shuts, and I'm squeezing on this, the pressure is getting really high in the ventricle. It should be greater than where? Where I'm in the pulmonary artery.
Oh.
So just getting it. So the pressure will be greater over here on the exiting side than on the opposite side of the pulmonary artery. So it would push open. But once that pressure changes, it reverses, and those valves will do what? Close, and so the blood is destined.
Oh, yeah. This is just copy something. So those semilunar valves have they don't open and close in of themselves, it's pressure only.
Pressure and muscle activity. Can you see how they're connected
have
to the
muscles?
muscles? They're
No,
connected
they're
to muscles.
semilunar.
Semilunar is correct, pressure only. No musculature
No
controlling.
muscle.
Right. So our valves are in our heart to prevent the blood from going back. So we wanted to go, right, we wanted to go in order and keep that pressure gradient established, keep our blood circulation going, keep us alive. And so if we kind of step away and look at it more broad, you can refer to figure 13.2 and look at how blood flows through the body. It's a closed circuit, so you can pick any point you want. I start when blood's already been to the systemic circuit, dropped off oxygen, and picked up what? Carbon dioxide. Now, not all of the oxygen should be unloaded, but some has, and we picked up some carbon dioxide. So the blood has been to the systemic circuit, and it needs to go to which side of the heart?
The left.
Let me back up. Before we get into details, our two functional circuits include the pulmonary circuit, which is your lungs, for what?
To get oxygen.
To pick up oxygen and
Release.
get rid of carbon dioxide. Our other functional circuit is the systemic circuit, which is where? The body. So everywhere else, not the lungs. For gas exchange, if it's endocrine glands, right, picking up hormones, dropping off, whatever, okay, we're body systems. The rest of the body receives systemic circuits. So if blood has been to the systemic circuit, dropped off some oxygen and whatever else, and picked up some carbon dioxide and whatever else, it's going to enter in the systemic capillaries and then into your veins, basically. The veins will dump into which vessels? The vena cava, which will drain into your right atrium. So blood will return from the systemic circuit to the right atrium. It will then drain into the right ventricle. I've got some arrows, so if you're going to follow along. The right ventricle will contract and propel blood out to the pulmonary circuit. And then here, we have a gas exchange. As the blood flows through, we said, what's going to happen?
We pick up oxygen and get rid of carbon dioxide.
Good. We're going to pick up oxygen, and we're going to drop off carbon dioxide. So carbon dioxide is going to diffuse into the lungs, and oxygen will diffuse from the lungs into the pulmonary circuit. Okay, so we're kind of refreshing. The blood will then transport from the pulmonary circuit to it would be the pulmonary vein, which would drain into the left atrium, which would then pump blood down into the left ventricle, which when the left ventricle contracts, pushes blood out, you guys are correct, into the aorta, which goes to our systemic circuit, to the rest of the body. Once in the systemic circuit, in the capillaries, you're going to see a reverse gas exchange where carbon dioxide is loaded into the blood and oxygen's unloaded into the tissues. And when that exchange happens, our proportions change and we're back where we started. It just keeps going with each beat.
So the carbon dioxide, the carbon CO2 blood does like a darker color because it doesn't have the oxygen, right?
Yeah, so in your textbook, you'll see red, purple, blue, but your blood is always which color? Red. In the textbooks, they'll be colored differently just so you can visually and quickly just kind of get some reference points. However, though, there is a difference in the tone of red depending on the amount of oxygen. So red blood that has high proportion of oxygen is going to be very bright, right? Kind of what you might think classic blood colored. However, if blood has gone to tissues and unloaded some oxygen, the hemoglobin, right, the iron's not bound, so it doesn't look as red, and so it looks more dark colored. So venous blood will look really maroon or burgundy compared to arterial blood, which should have a nice, brilliant, bright red. What else was added? So we shouldn't have blue colored blood. There are species that do, but they have a different metal that binds to the gases. What metal could turn blue? Copper. Do what? Okay, so there are some does anybody know an animal that has do what? Lizards?
I think we have red blood.
I don't know about lizards. Are there any herpetologists? Do what? The one I know of, and I'm sure there's others, the horseshoe crab, a little archaic looking thing, drain its blood is blue. So, but ours is not.
That's weird.
Good. Fun? All right. So back to the humans. Your book also makes a good acknowledgement about the heart. That the blood going through the heart does not meet the metabolic demands of the heart. It's just there passing through. The heart needs its own systemic circuit. It needs its own arteries, arterioles, capillaries, etc. Which you may be familiar with. You've had someone say, "I'm not a coronary," right? What they're joking about, they're talking about the systemic circuit to the heart being blocked or something like that. So the heart has its own systemic circuit. It doesn't happen to kind of.
So that's usually where all of that happens when people have heart problems in the heart's own arteries.
Yes. When someone when someone has, especially like acute heart attack, like myocardial, that would be the result of it. So if the blood vessels to the heart muscle, to the contractile cells of the muscle, and most famously the left ventricle, so we really want to get the blood to the rest of the body. If that blood supply is depleted, those tissues become necrotic. Their oxygen levels drop and they won't survive and they won't work. And so those cells will die. You might have an infarction.
Isn't it if you have like a heart problem, either something with the coronary arteries or it's a valve?
But there's many reasons why someone might have a problem with the heart. Could be a coronary artery problem. Could be a dietary problem. It could be a valve problem. It could be a pacemaker problem. Right? We keep naming every tissue in the heart and say there could be something wrong with that. But here in America, where our diets are top notch, right? We tend to fill our blood vessels with different types of lipids, which blocks all of our blood vessels, but we see to have the most noticeable problem when our heart stops receiving blood flow from the attack.
Kind of off the cuff, but have you heard of any benefits from extra virgin olive oil?
I'll briefly talk about it a little bit. The easiest way to say this, I had a great, I heard it yesterday actually, a great way to say this. The poison is not in the drug, it's in the dose. The poison's in the dose. Okay, so sure, olive oil's okay. Is it, if you eat olives, is it okay to go drink olive oil? No, because it's full of what? Fats. We should go, right? Is it good to eat soybeans? Yes. Is it good to eat only soybeans? No. Is it good to eat some meat? We're getting some moral, ethical thing here, but we're going to go with, we have to eat, sure. Is it good to eat meat? Yes. Is it good to eat only meat? No. So olive oil is just another thing that you need to keep balanced in the diet in small proportions like you would do with butter or something like that, because it's also still just a fat. So back on task, back to the heart. You guys identified properly, figure 13.6, about the difference in anatomy of the left ventricle and the right ventricle. You guys properly identified that the left ventricle has a much more muscle mass compared to the right, and that's because it has to propel the same stroke volume throughout the entire body circuit. So it's going to take more pressure to push that blood further. more resistance. Contrast it to the right ventricle, which has to push blood, I say it, next door to the lungs. So even though stroke volume is the same from each ventricle, the pressure needed to generate by each is different. And so therefore it's going to take more muscle mass to push that blood out.
Oh, that makes sense. Because it's like a smaller hole, and it's like whenever you take a hose and you put your thumb over the end, and it shoots out faster, rather than
The high pressure.
I just got that.
Okay, very good.
I just made the connection.
Very good. All right, so there are anatomical differences between the left and the right ventricle. And then we did have a bit of discussion already about the valves. So what you can do is review to figure 13.7, 13.8. And what it's identifying is that the AV valves and the semilunar valves will work in opposition, if you will, alternation. I don't know what the right word is. But when one is open, the other should be closed. And when the ventricles are relaxed and filling up, the AV valve should be open, and the semilunar valve should be closed. And then the reverse is true. When the ventricle contracts, the AV valves will close, and the semilunar valve should be opening up, pushing blood out. So when the ventricles are contracting, they're pushing blood out to each respective circuit. So we need to open that valve, that door, to the pulmonary or the systemic circuit. Again, those are figures 13.7 and 13.8 in your textbook. Are we okay to move on? Next thing I want to address is figure 13.9, reviewing the relationship of a pacemaker and the contractile cells. I know that we introduced it in the smooth muscle, but you're probably had more experience or familiarity in the heart. The heart has a set of cells which are called the pacemaker cells, and they're of course connected to the contractile cells. I'll use the word contractile cells. The proper word is myocardial, the heart muscle. But I say contractile so we can realize they contract. You're going to find contractile cells throughout the heart, in the atria and the ventricles. But the contractile cells of the left ventricle get the most fame, right? So that's the one that sends, that's the set of cells that send blood out to the entire systemic circuit. And I want you to be aware that the atria also contract, and so does the right ventricle. So the way that blood gets pumped in that fashion through our body in a coordinated activity is through the interconnectedness of all the cells in our heart. And it goes back to our familiar gap junctions. All right, so let's label this image. But like the big quotes, the pacemaker of the heart is an area of the heart called the sinoatrial node. It is the pacemaker for the heart. It's going to spontaneously, regularly what? Depolarize to threshold. So the pacemaker cells will spontaneously depolarize to threshold, which initiate action potentials. Action potentials are conducted to our contractile cells, which are shown here, labeled muscle cells. You can say myocardial cells. You can call them contractile cells. But the more orange, branchy-looking cells are the contractile cells of the heart. I have a couple more animations, like flashy. I'm going to show some flashes and arrows to illustrate action potentials, where they originate and how it's conducted. So hopefully this will help you understand the flow of the transfer. Between each cell is a specialized junction called an intercalated disc. Inter means what? Between, right? So collated, cross, right? Intercalated disc is the name of the junction between heart muscle cells. The intercalated disc, there are two primary types of membrane junctions, which we became familiar with in chapter 2 maybe, or somewhere around there. One type of junction that you'll find at your intercalated disc include things called desmosomes. You may recall those in that they're just basically interlaced proteins between two cells to distribute tension and prevent tearing. Another junction that you'll find a lot of at the intercalated disc are called gap junctions. And if we've been talking about these as well, we know that the gap junctions allow for ion transfer and action potential conduction.
Transfer and what?
Action potential conduction.
What did you say that?
Desmosomes prevent tearing. That's what they're trying to do. And what happens is the SA node is going to generate an action potential. This leaky. It's going to generate an action potential. I'm trying to show by the little flashiness. That is connected to our other cells by gap junctions. And so it'll just flow, right, from one cell to another, right through those gap junctions, right, and conduct, right, throughout the rest of the heart. What questions do you have over the terminology of using an intercalated disc, or the role of the gap junctions and the role of the desmosomes? If we have an idea of how all these cells are interconnected, we can look at the way that this would flow through the heart. First, turning to this image that I found, someone has labeled for you where the SA node is actually located. It's not something you can just look at a heart and, oh, there's the SA node. You would have to look at it with a microscope to really differentiate. And so if you look on the far right, the SA node is highlighted here. And you see how it looks maybe a little bit different than the muscle cell. You notice that where the muscle is, where my hand is, it's a little bit more densely colored or packed with protein when contrasted to the SA node. Okay? And the SA node is located on the right side, right above the right atrium, where the superior vena cava drains into the right atrium. Okay? That's where it's located. And it would be connected to all these muscle cells by what? Gap junctions.
I have a question. On the membrane junctions found at the intercalated disc, is it desmosomes and gap junctions, or would it be just, because by the picture I didn't know.
It would be both.
Okay, I don't know.
By the previous picture,
Yes.
the
So
previous
you
picture
have-
had both.
You have a gap junction, but it's being held together with the desmosomes?
Yes.
Okay.
Remember, gap junctions are just permeable. They're just a direct kind of tube, if you will, cytoplasmic connection. But the desmosomes both are expressed, and it wouldn't be just one of each.
Yeah, no,
What
So
are
yeah.
those, I'm sorry. Those brown wires, between the desmosomes. They made them look like Z.
This thing,
Yeah.
those are little protein filaments that touch and intertwine
Oh.
between. So inter, right, is between those intercellular junctions. So that's just the specialized proteins between them. So you would find both in between. And if we look at what happens, we know that the SA node is going to depolarize the fastest. Okay? And so it's going to generate our action potentials. So our little flashy light. What happens is these cells are connected, and then action potential is conducted not only down through the right atria, but across over to the left atrium. So action potential spread from one atrium over to the other through a specialized set of pathways called the interatrial pathway. That's a pathway between the atria. So they have the ability to conduct right to left. Before I get too far into the pathway, be aware that when this action potential spreads, the contractile cells will what? Contract. And so we're going to see contraction first with the atria, right? We're exciting the atria first. After it spreads from the SA node over to the left side of the heart, there's another pacemaker cell. So it's called the AV node, which is between the atrium and the ventricles. The AV node will conduct action potentials through a set of fibers shown here in the yellow called the AV bundle, or you may call it the bundle of His. The person named it. You may note that the bundle will then do what? And it branches, it splits into the right and left sides. And then it'll reach what's called the apex of the heart, the bottom, and you'll see the action potential spread up. From the bottom up, the outer portion of the ventricles. Through these fibers called Purkinje fibers. The terminal fibers there. So again, the electrical pathway, and then therefore the physical contraction, initiates after the SA node depolarizes the threshold, so that both atria will depolarize and contract simultaneously. Then the AV node, the AV node will generate an action potential down this thing called the bundle of His, which controls each the left and the right ventricles. But notice the contraction will actually start. Do I know the name of this thing in the middle?
Septum.
It's called the septum. You would call it the interventricular septum. So the contraction will start in the septum, go towards the base of the heart, and then back up. And so we'll depolarize middle, bottom, outer, up. So our heart will contract that way. What questions do you have about this pathway of excitation through the heart? Okay with it? All right. This is what we're reviewing in lab this week. And because we can't go a day without saying homeostasis or something like that, so we're going to talk about our action potentials. And it looks like I have an image to show first. So before we look at the action potential, don't change your page yet. Still looking here. Histologically, looking at cross-section of the heart, we have different layers. And if you recall, we were just looking at those yellow fibers, talk to the nodes and the bundles and Purkinje fibers. They're specialized cardiac tissue to conduct action potentials. Right? So all these things, the SA node, the internodal pathway, the bundle, and all those things, although they're cardiac tissue, they don't do what? They don't contract, but they do conduct action potential. So they're kind of like what? They're kind of like nerves. They're like nerves, but they're not nerves. They're heart cells that conduct action potentials. So the bundle of His, the internodal pathway, the left and the right branches are just conducting cells. They don't generate, they don't initiate the action potential, but they conduct it through the heart. So all those things that you listed are just conducting, not contracting. Does that make sense? They're like little electrical wires in the heart. Heart muscle cells.
Oh, the SA as well.
The SA node does conduct this, it's properly called the pacemaker, so it starts that pathway. Did I help or confuse? Good. And this image should back that up. We can see highlighted in area 1 some of those conducting cells called Purkinje fibers. if you look at it, again, it's kind of hard to see in this room. Want to black out? If you can see the difference. Okay. Maybe I will see the anatomical difference now. Not even knowing histology, you can see this area looks lighter, and this area, these areas look darker, right? And we learned that with the light microscope, areas that are more dark have more somethings in them. The muscle cells have more thin and thick filaments. The conducting cells, it's not really apparent, but maybe they do. Their job is to conduct, not to contract. Lights on. So if we look at the electrical activity, because we can't get away from those pacemakers, the action potential is well characterized in the pacemaker cells contrasted to the myocardial cells. So I'm able to answer the question better for cardiac muscle than you were asking about smooth muscle action potentials. First thing is, what type of tissue would do this regularly? We would call this what type of cells? We said pacemakers. So highlight it, circle it, or whatever. This would be in the nodes, the SA node or the AV node. Action potentials are real regular in our pacemaker cells. And our pacemaker cells depolarize regularly to our good friend calcium. Calcium in the pacemaker cells leaks in to bring the pacemaker cells to their threshold. This is different than what we've seen in our other tissues. Depolarization usually moving on sodium pretty much all semester, right? The pacemaker cells depolarize thanks to calcium flexing in. Then we'll reach threshold and then rapidly depolarize. So the takeaway is our pacemaker cells are leaky to calcium. They continually bring up the threshold, generate an action potential, and then go back. It's continuous. The action potential of the myocardial or the contractile cells is very much different. Okay, so it's going to look really, right? Let me finish labeling that. So our familiar depolarization, repolarization happening during our pacemaker cells. To contrast the action potential of a pacemaker cell to a contractile or myocardial cell, it's going to look very different from what we've seen before. Palpate the enthusiasm in the room. All right. Sorry. Get the show on this. So it's going to repolarize, depolarize after threshold, and repolarize like we're familiar with. Okay, for the pacemaker cells. What's unique is getting the threshold through our calcium influx. Okay, that's the pacemaker cells. But the bulk of the heart, 99% of the rest of the heart, are contractile cells. Okay? Those contractile cells, the myocardial cells, look at their action potential. Looks very different, right? They're going to depolarize the threshold back to our familiar sodium ion. So the contractile cells, when a stimulus is present, they will depolarize, open up, let sodium into the cell. And then they do something really weird. As they begin to repolarize, letting potassium out, so it starts out a little bit similar, and then we have this weird, I don't know if it's weird, but I would use it, it's not nice, this unique, right, plateau phase that we haven't seen before. And so we kind of max out. Does anybody know what happens during that plateau phase? You read ahead? You did that? Plateau phase? Ah, that's really sad. I'm just kidding. Calcium leaks in. So sodium in, calcium out a little bit, and then we let calcium in. So we keep it excited. Excited, start to go back to rest, and then keep it excited for a short period of time. And that's called a plateau.
So it's not like, right here, the calcium and the sodium point right here.
Mm-hmm.
Calcium and the sodium are right here, but here is just the sodium and then calcium.
Yes. The question is, when does calcium come into play in the contractile cells? And it's uniquely during that plateau phase, contributing to the action potential. The reason your contractile cells have this plateau phase, let's go ahead and acknowledge it now, is to prevent summation. That plateau phase prevents summation of your muscle cell contractions. Why is that important?
So that you get all the blood out of whatever it's going from.
So she said, so we get all the blood out, or all the blood in, or right, whatever. So the heart muscle contracts and then relaxes. We don't want it to contract and then contract some more and then contract some more and then tetanus or something, right? We don't want that in the heart. That was okay in the skeletal muscle. We're trying to move something. But in the heart, if it stops pumping, it's not working. So if it maintains a contraction for a long period of time, someone's in cardiac failure, and they're in the middle of a heart attack. We don't want that, and so we can protect that, prevent that from happening by maintaining that excited state. Okay? Once it's excited, it can't receive a new stimulus. Does that make some sense? It's in the middle of dealing with one, it can't deal with another while it's in this excited state. Then after the plateau phase, we'll repolarize with our familiar potassium ion.
So is the first part like a partial repolarization?
Yeah, and really the whole thing is a repolarization, but it stalls. Stalls out during what's called the plateau when calcium now comes in to the contractile cells. Good. We have about 5 minutes. I think we can get it covered. So let's go ahead and keep moving. The next thing is figure 13-14, excitation, contraction, coupling. This will be a little easier to work through skeletal muscle and we work through smooth muscle. And if you recall, the cardiac muscle is kind of a conglomerate of the two. And the first thing is, in this image, 13-14 in your textbook, this is one cell and another. This is not, the green thing is not a T-tubule. Okay? On the far left, one cell. On the far right, another cell. So be sure to label that. So this is not a T-tubule. These are two different cells connected by a gap junction. And so this first cell, it could be the SA node. It could be one of the Purkinje fibers. It could be another contractile cell, but it's connected to this contractile cell by gap junction. So whatever cell it is, we're going to conduct that action potential from one cell to another. If you recall, once a myocardial cell gets excited, we're going to let which ion into the cell to initiate the contraction? Calcium. It's going to flood in from the extracellular fluid. So generate an action potential, and then calcium floods into the cell from the extracellular fluid. That triggers calcium from the SR, the sarcoplasmic reticulum. Because our cardiac muscle cells have thin and thick filaments, calcium will bind to troponin, which pulls back tropomyosin, which allows for myosin to bind with its binding sites on actin. So the actual cross-bridge cycling is just like skeletal muscle. But the initial source of calcium is like smooth muscle. Is it easier by the time we get to the third one? Hopefully. Okay, I'll take that statement back. Again, these events are summarized and certainly much more wordy if you refer to figure 13-14. But the point is, wherever the source of excitation is, depolarize the threshold to let which ion? Calcium in from the extracellular fluid, which causes the SR, sarcoplasmic reticulum, to release its calcium into the cytoplasm to bind with troponin to allow for contraction, that cross-bridge cycle. When the cell is repolarized back to resting membrane potential, calcium goes back to the extracellular fluid and into the SR. It looks like we won't have time to talk about the EKG, so you won't be responsible for the EKG on the test. And we'll review. We'll pick up after the test on Tuesday. On Thursday, we'll pick up with the EKG, and we'll move right into blood vessels next Thursday. We'll conclude our discussion here.